Photosynthesis is the process of converting light energy into chemical energy by green plants and certain other organisms. The most common and critical type of photosynthesis happens in chlorophyll-containing plants, algae, and cyanobacteria. We'll tackle what chlorophyll is later. Light energy is captured and used to convert water, carbon dioxide, and minerals into oxygen and energy-rich organic compounds during photosynthesis in these organisms.
You know, photosynthesis was first discovered by the Dutch-born British physician and scientist John Ingenhousz, who was born on 1730 and died in 1799. He discovered photosynthesis sometime on or before 1779. This discovery was published as the results of an ingenious study on the chemical effects of plant physiology, experiments upon vegetables, discovering their great power of purifying the common air in sunshine and of injuring it in the shade and at night.
Photosynthesis is very crucial for the survival of all living organisms. Hey Broward, do you mind explaining the importance of photosynthesis?
Easy peasy. Photosynthesis is important to living organisms because it is the number one source of oxygen in the atmosphere. We learned that photosynthesis is plants taking in water, carbon dioxide, and light to make sugar and oxygen. And this is important because all living things need oxygen to survive. Photosynthesis also helps in balancing atmospheric carbon dioxide and oxygen, and this is because the carbon dioxide in the atmosphere is absorbed by green plants to manufacture carbon compounds. This will also help reduce or prevent greenhouse effects.
Thank you for that, Broward. Now, I gathered all your questions and we are going to answer all of them today. First, where does photosynthesis occur?
It occurs in autotrophs, which are organisms that can produce their own food that contain the green pigment chlorophyll. This would include the entire plant kingdom, half of the protista, the algae, and half the monomer kingdom, the blue-green algae. Plant photosynthesis occurs in leaves and green plants within specialized cell structures called chloroplasts. One plant leaf is composed of tens of thousands of cells, and each cell contains 40 to 50 chloroplasts. The chloroplast, an oval-shaped structure, is divided by membranes into numerous disc-shaped compartments. These disc-like compartments, called thylakoids, are arranged vertically in the chloroplast like a stack of plates or pancakes. A stack of thylakoids is called a granin, plural granum. The grana lies suspended in a fluid known as stroma. Embedded in the membrane of the thylakoid are hundreds of molecules of chlorophyll, a light-trapping pigment required for photosynthesis.
Well explained, Jillian. Next one. Why are most plants green? I was wondering about that the other day.
Great question! We all agree that we were mesmerized by the exquisite colors of the plants. But did you know it's because of photosynthetic pigments? Now, the photosynthetic pigment is a pigment that is present to absorb the light energy required for photosynthesis in chloroplast. Because they interact with light to absorb only certain wavelengths, pigments are useful to plants and other autotrophs. In plants, algae, and cyanobacteria. Pigments are the means by which the energy of sunlight is captured for photosynthesis. However, since each pigment reacts with only a narrow range of the spectrum, there is usually a need to produce several kinds of pigments, each of a different color to capture more of the sun's energy. Pigments are chemical compounds which reflect only certain wavelengths of visible light. This makes them appear colorful. Flowers, corals, and even animal skin contain pigments which give them their colors. More important than their reflection of light is the ability of pigments to absorb certain wavelengths.
I bet we are all curious how photosynthesis really works. Are you ready? Let's go! There are two stages of photosynthesis, namely the light-dependent reaction and the light-independent reaction, or the Calvin cycle.
The first stage of photosynthesis is the light-dependent reaction. As its name states, it uses light energy to make two molecules, the energy storage molecule ATP and the reduced electron carrier NADPH, which are needed for the next stage. There are three components that must meet in the leaf cell's chloroplast and its thylakoid discs, which are flattened sacs of membrane, for photosynthesis to occur. Photosynthesis begins when sunlight strikes the leaf and the photon of light energy travels until it reaches a molecule of chlorophyll, which are located in the thylakoid membranes. The photon causes an electron in the chlorophyll to become excited. The energy given to the electron allows it to break free from an atom of the chlorophyll molecule. Chlorophyll is therefore said to donate an electron. To replace the electron in the chlorophyll, a molecule of water is split. This splitting releases an electron and results in the formation of oxygen and hydrogen ions in the thylakoid space. Technically, each breaking of a water molecule releases a pair of electrons and therefore can replace two donated electrons. The activated electrons then go through the electron transport chain and in the process leads to the accumulation of the hydrogen ions inside the thylakoid membrane. The thylakoid membranes are the disc-like structures in the chloroplast that contain chlorophyll, generating a proton gradient. As the protons go down their concentration gradient through the ATP synthase, ADP and P come together to form ATP. The adenosine triphosphate, or the ATP, is an energy molecule that cells exchange to release energy. The electrons going through the electron transport chain eventually end up combining with NADP+ to form NADPH, another energy-rich molecule that will be used later. Thus, the result of the light reaction is the production of oxygen released from the leaves, and ATP and NADPH, which will be needed for the later steps. The main difference between ATP and NADPH is the amount of energy molecules they release.
To summarize, photosynthesis begins with the light reactions. It is during these reactions that the energy from sunlight is absorbed by the pigment chlorophyll in the thylakoid membranes of the chloroplast. The energy is then temporarily transferred to two molecules, ATP or adenosine triphosphate and NADPH or nicotinamide adenine dinucleotide phosphate hydrogen, which are used in the second stage of photosynthesis. ATP and NADPH are generated by two electron transport chains. During the light reactions, water is used and oxygen is produced. These reactions can only occur during daylight as the process needs sunlight to begin.
The second stage of photosynthesis is the light-independent reaction, or better known as Calvin cycle. All living organisms here on Earth, including you, are a carbon-based life form. This only means that the complex molecules of your amazing body are built on carbon backbones. The atoms of carbon in your body were once part of carbon dioxide molecules in the air. Carbon atoms end up inside you thanks to the second stage of photosynthesis known as the Calvin cycle. The Calvin cycle is the second stage of photosynthesis. It is the process where autotrophs are used to create nutrients from sunlight and carbon dioxide. The process was first identified by American biochemist Dr. Melvin Calvin in 1957. Plants and algae are examples of autotrophs, and they use carbon dioxide from the air and convert it into glucose, which is a sugar, the food they need to grow. This is the process where the chemical energy is stored in the ATP and NADPH. The products of light reactions are used to convert CO2 to glucose. This process happens in the stroma of the chloroplast.
Now, plants depend on Calvin cycle in order to produce food. But don't you ask how they do it? Well, to answer your question, Calvin cycle undergoes 3 reactions and 6 turns in order to produce their foods. Since carbohydrate molecules have 6 carbon atoms. These 3 main stages in a Calvin cycle are the carbon fixation, reduction, and the regeneration of the starting molecule. So to understand these reactions, let us take a closer look to these processes for us to understand how it works.
The first stage in the Calvin cycle is called carbon fixation. This involves the merging of CO2 into an organic material. In this phase, the CO2 molecule is attached to a 5-carbon sugar molecule called ribulose biphosphate or RUBP. This attachment was made possible by the enzyme called Rubisco or RUBP carboxylase. This enzyme is believed to be the most abundant protein in the chloroplast. The resulting product of carbon fixation is a 6-carbon sugar, which is extremely unstable and immediately splits in half. The split forms 2 molecules called 3-phosphoglycerate.
In the second phase, we have reduction. This phase gets its name because NADPH donates electrons to or reduces a 3-carbon intermediate to make G3P. In the reduction phase, the 3 phosphoglycerides are going to be phosphorylated by the ATP coming from the light reaction. There are 6 molecules of 3 phosphoglycerides, so the cycle also needs 6 ATPs. The ATPs will give one of their phosphate group to the 3-phosphoglycerate via an enzyme called 3-phosphoglycerate kinase, forming 6 molecules of 1,3-diphosphoglycerate, meaning they now have 2 phosphate groups attached to their molecules, more specifically at the first and third carbon molecules. In this stage of reduction phase, ADP is produced as a byproduct. The 6 molecules of 1,3-diphosphoglycerate are reduced via the enzyme called NADP glyceraldehyde 3-phosphate, which oxidizes the 6 NADPH molecules, which comes from the light reaction and gives the electrons to the 1,3-phosphoglycerate, and at the end there will be a production of 6 molecules of glyceraldehyde 3-phosphate. Again, in the same stage, NADP+ is also produced as a byproduct.
After being phosphorylated and reduced, the 6 G3Ps are now produced. 5 molecules of G3Ps are recycled to give 3 molecules of RUBP to contribute and run the carbon fixation again. Thus, these 5 G3Ps will proceed to the next phase. The remaining 1 molecule of G3P exits the cycle to make glucose later on, and is used to synthesize sucrose or starch. A G3P molecule contains 3 fixed carbon atoms, so it takes 2 G3Ps to build a 6-carbon glucose molecule. Hence, it will take 6 turns or 2 revolutions of the cycle to produce 1 molecule of glucose. After the 6 turns or 2 revolutions of the cycle, the 2-glyceraldehyde 3-phosphate then combine to form 6-carbon sugar. They could either be glucose or fructose. Eventually, glucose and fructose can be combined to form sucrose. Starch is formed when glucose is connected in chains. The byproducts formed during the Calvin cycle, which is ADP and NADP+, will then be brought back to the thylakoid membrane and then will enter again the light reactions. Inside the thylakoid, they will be recharged with energy and become ATP and NADPH.
The third and the final phase of the Calvin cycle is called the regeneration of RUBP. From the name itself, it simply states that RUBP is being regenerated to continue the cycle. But to be more specific, the 5 molecules of G3Ps who continue their journey towards the cycle will undergo complex enzymatic reactions to produce 3 molecules of ribulose biphosphate or RUBP. Since we have 5 molecules of G3P that contains 3 carbons, it means that we have 15 carbons, and these 15 carbons will just reassemble and form 3 molecules of RUBP, which contains 5 carbons each. And these 3 molecules of RUBP are now ready to be fixed by 3 molecules of carbon dioxide and enters again the cycle. But take note, before the reassemble occurs, 3 ATP molecules were produced, and because of the produced ATP, this allows the Calvin cycle to continue.
Calvin cycle is very significant to every living thing on Earth since we depend on it. Plants depend on the Calvin cycle for energy and food. Other organisms like herbivores also depend on it indirectly because they are fed on plants. Even animals that eat other organisms, such as carnivore, depend on the Calvin cycle. Without it, we wouldn't have the food, energy, and nutrients we need to survive.
To summarize, the Calvin cycle or the dark reactions has 3 phases, namely Phase 1, carbon fixation, Phase 2, reduction, and Phase 3, regeneration of RUBP. In Phase 1, the carbon dioxide are merged to make an organic material. Carbon dioxide attaches to RUBP with the help of Rubisco. The result is a 6-carbon sugar that splits in half to form 2 molecules of a 3-phosphoglycerate. In Phase 2, a phosphate group from ATP attaches to each 3-phosphoglycerate forming 1,3-phosphoglycerate, and ATPase reduces 1,3-diphosphoglycerate to produce G3P. A single Calvin cycle will produce 6 G3Ps, but only one of them will be packaged for the use by the cell. Hence, it takes 6 turns or 2 revolutions to make glucose. ADP and NAD+ will be brought back to the thylakoid membrane and become ATP and NADPH. Finally, in Phase 3, the recycled 5 G3Ps will undergo enzymatic reactions to make 3 molecules of RUBP, which is needed to continue the Calvin cycle, which has sustained and nourished the Triceratops, both herbivorous and carnivorous. Remember, to make 1 G3P molecule, the chloroplast needs 3 molecules of carbon dioxide, 9 molecules of ATP, and 6 molecules of NADPH. Some important points to know about the Calvin cycle is that the result is not the 6-carbon sugar glucose, which will be formed later on, but the 3-carbon sugar known as G3P. The cycle also needs to spin 3 times to make 1 molecule of G3P from 3 molecules of carbon dioxide. The cycle also needs to revolve 2 times to make 1 glucose molecule.
In conclusion, photosynthesis is the process by which plants, some bacteria, and some protistants use the energy from sunlight to produce sugar, which cellular respiration converts to ATP, the fuel used by all living things. The conversion of unusable energy to usable chemical energy is associated with the actions of the green pigment chlorophyll. Most of the time, the photosynthetic process uses water and releases oxygen that we absolutely must have to stay alive. And yes, the food as well. That's all for now, and we remind you to always
Keep learning. Bye.